So let's start with that. Fine. So we were here in endomembrane system. Endomembrane system, those organelle, those cell organelle which are made up of membrane, right? And the endomembrane means the membrane, the organelle which are made up of membrane, and they are inside the cell. So those organelle are called endomembrane system. Out of endomembrane system, I have already discussed in the last class. This endomembrane system, we discussed endoplasmic reticulum, Golgi complex, and lysosome, right? Okay. So lysosome are of two types. Endoplasmic reticulum are of two types. Martin, can you check that had I dictated it in last class or not? Martin, can you check with your notebook? Am I audible to you, Martin? Hello, Martin. You are not audible to me.
Sorry, sir. I was in the washroom. Yeah,
Okay.
hello.
Yes, yes. Now you are audible. So, had we done with it?
We were going to start. We finished the rough year and smooth year.
We have finished the endoplasmic reticulum.
Mm.
Okay.
Yes, yes. Sir.
So we will start with the next organelle.
Yeah, yeah.
Fine, Golgi complex. Okay, thank you. So let's start with the Golgi complex. So look, Golgi complex are also known as the Dalton complex, right? That is the same thing. This is the structure of Golgi complex. It is made up of some component. So the first component is called cisternae. So these macaroni kind of the things, they are called larger macaroni, smaller, more smaller, more smaller, more smaller. So these things are called, these structures are called cisternae, right? The space is inside the cisternae is called lumen, right? Then there are these spherical objects, these spherical structures are called vesicle. So basically, Golgi complex is made up of cisternae and some vesicle. One very important thing I want to tell you, that Golgi complex have two faces. Look, this concave safe face, it is called cis face, right? Try to understand. This concave side face is called cis face, and this convex kind of the face is called trans face, right? Now, cis face and trans face, this is the only organelle which have polarity, right? Which have two poles, cis face and trans face, right? One very interesting thing, the cis phase will always face towards nucleus, and trans phase will always face towards the cell membrane, right? That's why we call this is the polar organelle, right? This is the organelle which maintain polarity. What polarity? One pole will be cis phase, another pole will be trans phase. Cis phase will always be towards nucleus, and trans phase will be away from nucleus, means towards plasma membrane, right? Then look, what is the function of this structure, or what is the function of this organelle? The main and the primary function of this organelle is packing of protein. It do the packaging of protein. So it take the protein, right? It take the protein from the endoplasmic reticulum, rough endoplasmic reticulum, and then modify them and then pack them. And after packing them, it release three kind of the vesicle. This it just look, the protein enter from here, and when it come out from the here, what will happen? It will come out as a three vesicle. These vesicle are of three type of vesicle. After packing them, it makes three kind of vesicle, A, B, and C. A kind of vesicle, they fuses with the plasma membrane and provide them protein, right? Why? Because plasma membrane is made up of two things. What? Lipid and protein. We have already discussed. Plasma membrane is made up of bilayer of lipid and protein. So protein supply is done from endoplasmic reticulum, right? Formation of plasma membrane means formation of new plasma membrane is called biosynthesis, right? So biosynthesis is the process in which formation of plasma membrane takes place.
Yes, sir.
For biosynthesis process, it requires lipid and protein. Why? Because this is made up of the lipid and protein. These are the component of plasma membrane. The protein it will get from the Golgi, and lipid it will get from the smooth endoplasmic reticulum, right? Second kind of vesicle are released from the cell, they make enzyme. Third kind of the vesicle, they live inside the cell in the circular vesicle, spherical vesicular form, and they form lysosome. So formation of lysosome also takes from Golgi complex. So there are three functions of Golgi complex. This was discovered by Camillo Golgi, right? Name of the scientist is Camillo Golgi. Give me just a minute, just 1 minute. Okay, so it was discovered by Camillo Golgi, right? And where they are not found, they are not found in the nerve. Sorry, they have been discovered. They have been discovered in the nerve cell of brine owl and cat, right? This is the organism where we discovered the Camillo Golgi discovered the Golgi complex. They are not found in sieve tube of plant, sperm of pteridophyte, and RBC of human. So basically, in some books, some notes, and some videos, it is said that Golgi complex are not found in the plant. That is wrong. Basically, this kind of the Golgi complex are found in the plant, and we give them different name. In plant, we call them dictyosome. Basically, they are not that much developed or organized like this way, right? They are not organized. They are found in the form of these vesicles, right? These cisternae. So cisternae are not arranged like in this way. They are just cisternae are for, so unconsolidated units are found. So they are called dictyosome. This is statement that if I say that Golgi complex are not found in the plant, this is completely wrong statement, right? The correct statement is that Golgi complex are found in the plant. They are called dictyosome over there. They are not well organized. They are organized in the form of unconnected unit of cisternae, right? 3 to 20 cisternae are found in per Golgi complex, right? Is that clear? Golgi complex clear?
Yes, sir.
Okay, please draw the diagram quickly. It's very simple. Quick. Then I will dictate for you. Done? Ilam? Martin?
Yes, sir, done.
Okay. Martin, done?
Yeah, yes, sir.
Okay. Please start noting now. Quick. Uh, Golgi complex are also known as Dalton complex. Dalton complex. Full stop. It was discovered by Camillo Golgi. It was discovered by Camillo Golgi in 1898. Camillo Golgi in 1898. Full stop. He discovered it. In the nerve cell of. He discovered it in the nerve cells of brain owl and cat. Brain owl and cat. Full stop. It is next point. It is present in all eukaryotic cell, except sieve tube of plant. It is the sieve tube of plant, comma, sperm of bryophyte, and pteridophyte, and RBC of human. Full stop. Next point. In plant cell, Golgi apparatus is formed of a number of unconnected unit, called dictyosome. It is written over here, called dictyosome. Full stop. The shape and size of, next point, the shape and size of Golgi complex, are not fixed. Next point. Golgi complex are made up of 4 to 8 membrane bound cisternae. Generally,
So 4 to 8 membrane bound.
Cisternae. It is written over. Look, cisternae.
Okay, sir.
Generally, but they may be made up of 3 to 20 cisternae. 3 to 20 cisternae. Normally 4 to 8, but minimum 3 and maximum can be 20. Now next. The function of Golgi complex is packaging of protein, and sending them out of cell full stop. Next point. Formation of lysosome takes place through Golgi complex. Next point. Golgi complex provide protein during formation of plasma membrane. Right. So this was all about the Golgi complex. Is it clear? If you have any doubt, please ask me. Crystal clear to both of you?
Yes, sir.
Okay. Now the next is lysosome. So same. First, I would like to explain it for you. I have already told you that formation of lysosome takes place from Golgi complex, right? Lysosome are also known as the suicide bag. Why they are called suicide bag? I will tell you. This is the important question for you. Why the lysosome are called suicide bag? Can you tell me why they are called suicide bag? Any one of you?
Sir, because they kill the cells or something.
Because they kill the cell. How they kill the cell? That's why that's what I want to know. Because their function is not killing the cell. Their function is digestion.
So when the cell gets damaged.
Sorry, they?
Sir, when the cell gets damaged.
Yeah, in cell which damage, they also kill that cell. Look, basically, try to understand. First, very important thing that this is the organelle which is made up of single membrane system, right? So it is made up of single membrane, not double membrane. Normally, most of like mitochondria, chloroplast, and all that they are made up of double membrane, outer and inner membrane, but it is made up of single membrane. First thing. Second important thing, this is very important. You need to remember it. Basically, inside this membrane, the enzyme which are filled, they are called strong hydrolytic enzyme, right? Here I have written a strong hydrolytic enzyme. So these strong hydrolytic enzyme are digestive enzyme. They are very strong enzyme. If accidentally lysosome rupture, due to rupture of lysosome, these enzyme come out in the protoplasm. They digest complete protoplasm and cell die. That's why they are called suicide bag. But normally they do not help in suicide or killing of the cell. So normally what happen? Normally they take the food, complex food material, they break down into smaller components, simpler components. So their basic function is digestive, right? Digestion of the complex food. 50 different type of enzymes are found. They are called suicide bag. And how does they maintain the very low pH inside? They have the hydrogen channel. So hydrogen channels are present. They keep on sending hydrogen inside the lysosome. That's why they have. That's how they maintain the strong acidic environment inside there, right? They are called scavenger of cell. So why they are called scavenger of cell? Basically, if any cell organelle become old, die, so they clean the cell and they eat and digest those cell organelle like mitochondria, chloroplast, and all that. So those cell organelle, if any cell organelle die, they eat and clean the cell. That's why they are called scavenger of cell. They were discovered by Christian Didue, right? Who discovered it? Fine. The pH value of lysosome, the enzyme inside the lysosome, is 4 to 5. They are found in all the animal cell except RBC. Plant and fungi, the function is taken away by the vacuole. So they are not found in the plant and fungi because their function is taken away by vacuoles. And they have single membrane, strong hydrolytic enzyme. These are digestive enzymes. Proteins are present, right? And the material inside is semi-crystalline material, clear? And diameter is 0.2 to 0.8 micrometer. Okay. So how do they digest? That I am going to share with you. You don't require this one, but let me share with you. Basically, like amoeba-like organism animal, they eat the some food particle. They are eating the food particle. Lysosome are present. Lysosome containing digestive enzyme. They are also present. Uh, they make the vacuole called food vacuole. This food vacuole fuses with lysosome. Lysosome enzyme work on this food. This is called secondary lysosome, right? The main lysosome is called primary lysosome. When it fuses with the vacuole, it is called secondary lysosome. After that, they digest the food material, and after digesting, they break down into small particle, and it is removed. And these waste particles are removed away from this one, right? So that's how they help in digestion of the food. So do one thing, please draw this diagram and write down only single membrane, strong hydrolytic enzyme, digest, and then protein, and that's it. Draw this diagram quickly. It's a very simple diagram. Then I will dictate each and everything that I have told you. Done.
Yes, sir.
Okay. So please start noting now. First one. They were discovered by a Belgian scientist. He was from Belgium, right? They were discovered by a Belgian scientist, Christian Didue. It is written Christian Didue in Christian Didue in 1955. In 1955. Full stop. They are bounded by a single membrane and contain, they are bounded by a single membrane and contain a strong hydrolytic enzyme. A strong hydrolytic enzyme. Full stop. The enzymes are present in the form of semi-crystalline granules. Full stop.
Can you repeat?
The enzymes are present in semi-crystalline granules. Full stop. About, next point, about 50 enzymes, about 50 enzymes have been recorded, about 50 enzymes have been recorded in them. Next point. They have a strong hydrolytic enzyme. They have a strong hydrolytic enzyme with a low pH value, with a low pH value 4 to 5. Full stop. The acidic condition are maintained. The acidic conditions are maintained inside lysosome by pumping hydrogen ion inside. Right. The lysosome are also called suicide bag of cell. Underline this statement. Lysosome are also called suicide bag of cell. Full stop. If the membrane happens to get broken, if the membrane happens to get broken, they will digest all the cell organelle. They will digest all the cell organelle and cell will die. Next point. They are also known as scavenger of cell. They are also known as scavenger of cell because they eat and digest They eat and digest old or dead. They eat and digest old or dead cell organelle. Full stop. Next point. Their main function is digestion of complex organic food. Their main function is digestion of complex organic food, and they break down them into simple component. Next point. They are found in all animal cell except RBC. Next one. In plant and fungi, their function is taken away by, in plant and fungi, their function is taken away by their function is taken away by vacuoles. That's it.
Sir, can you explain the last point?
Yeah. In plant and fungi, their function is taken out by vacuole, because you know the function of lysosome is digestion of what? Digestion of food, right? So these function and they have a strong hydrolytic enzyme. The same function is done by vacuole in plant and animal, because the plant vacuoles are not found in animals. Vacuoles are found in plant, right, and fungi.
Okay, sir.
Okay, so they are no more required in the plant. That's why they are not found in plant. They are found only in animal cell. If you want, you can note it down. They are found only in animal cell, not in plants. Is that clear, Martin?
Yeah, yes, sir.
Okay. The next one is a vacuole. Right. So look, every plant cell have the biggest organelle of plant cell is vacuole. Let me tell you, when the plant cell is not mature, when the plant cell is immature, so there will be very small vacuole. When the plant cell will get mature, what will happen? Those small vacuole will fuse and they make a big vacuole, that is called central vacuole, right? So that is called central vacuole. So every mature plant cell have central vacuole. Vacuole is the largest organelle. It takes lots of space. So you can see here, in every plant cell, in any plant cell, you won't found nucleus in this at the center. Why? Because vacuole take lots of space. Vacuole push the nucleus and protoplasm aside, right? So vacuole put the nucleus and protoplasm at peripheral part. So you can see nucleus and protoplasm at the peripheral part, right? And they are kept aside. So this is the nucleus, this is the protoplasm, this is the vacuole. What is the function of vacuole? Look, one very important thing which I want to pinpoint over here. The membrane, this is the only membrane which have some different name, right? Else every cell organelle have, like we call that inner membrane, outer membrane, or simple membrane. But yes, the membrane which surround the vacuole is called tonoplast, right? Okay, this is selectively permeable. Inside the membrane, there is a water, there is a pigment, there is a mineral, there is a uh, uh, some other component, right? And vacuole maintain the shape of cell. Vacuole store the lots of this water and other things, right? So that is the function of vacuole. This is the structure of vacuole. So do one thing, make a cell, a big vacuole, right? Cell wall, plant. So don't make all these things. Nucleus, protoplasm, right? And cell wall, plasma membrane, and vacuole. That's what you have to draw. Please, quickly draw it and let me know.
Done, sir.
Done? Okay, fine. Please start counting now. Vacuole. Vacuoles are believed to be formed by vacuoles are believed to be formed by endoplasmic reticulum, ER. Vacuoles are believed to be formed by endoplasmic reticulum. Full stop. Next point. They are filled with a fluid. They are filled with a fluid called sap, S-A-P, called sap. Full stop. The membrane of vacuole is called tonoplast, and it is selectively permeable. Next point. A mature plant cell have a mature plant cell have a single large central vacuole which occupy a mature plant cell have single large central vacuole which occupy 90% of volume. Next point. The function of vacuole, the function of vacuole is that they maintain the shape of cell. The function of vacuole is that they maintain the shape of cell. Full stop. They may store reserve food. They may store reserve food. Full stop. They store and concentrate waste product. They store and concentrate waste product. Next point. They store water, comma, mineral and pigment. They store water, mineral and pigment like anthocyanin. That's it for vacuole. And one more thing. This is the largest organelle of plant cell. This is the largest organelle of plant cell. Full stop. I think it should be clear to both of you.
Yes, sir.
Okay. Lysosome is very important. Why they are called suicide bag? That is the question. That will be 2 or 3 marker question for the school examination. Keep on noting down and keep on taking on your NCRT. Take down that in NCRT. What I am telling that. Look, in mitochondria, this diagram, the first important question, draw the well-level diagram of mitochondria. If you get the diagram-based question, then the question can be, they can ask you 3 marker or 4 marker question. Draw the well-level diagram of mitochondria. So diagram of mitochondria is very important. Right? Now the mitochondria itself is very important. So before telling you anything, let me let you know that what is the function of mitochondria, what exactly the mitochondria is, what exactly it do. So mitochondria is called powerhouse of cell. Why? Because it generates the power. How it generates the power? Basically, the glucose comes inside the mitochondria. It burns the glucose inside mitochondria, and due to burning of glucose, it generates energy. And energy is generated in the form of what? Tell me. In what form energy is generated from mitochondria? Both of you, Elam and Martin, tell me.
ATP.
Very good. ATP, right? ATP is called adenosine triphosphate. So ATP is also known as the currency of cell, right? So what happens? Suppose that in every cell of our body there is a mitochondria. The cells which are more active in our body, they will have more mitochondria, like cell of heart, cells of liver, right? Cells of neurons, they will have more mitochondria. The cells which are very less active, they will have very less mitochondria, like skin cells. They are not that much active. There will be less mitochondria. So the number of mitochondria vary, but it depends on the activity of cell. If cell is more active, there will be more number of mitochondria. If cell is less active, there will be less number of mitochondria. Mitochondria take the food in the form of glucose. It takes glucose inside and then it burns it. After burning, they produce ATP. ATP gets stored in our body. Whenever the cells or body require energy, ATP breakdown and they provide us energy. That's why ATP are also known as the currency of energy currency of cell. Okay. New mitochondria arise from pre-existing mitochondria. Means mitochondria have capability to divide. It divide and form new mitochondria. Okay. Now come to the structure. This was the function of mitochondria. Come to the structural part. That is very important. Look, mitochondria have two membranes, inner membrane and outer membrane. You can see this is the inner membrane and this is the inner membrane and this is the outer membrane. So there are two membranes. Between these two membranes, there is a space. Now, inner membrane is folded innerwards, right? They have finger-like structure. So inner membrane is folded and it make a finger-like structure. These finger-like structure are called cristae, right? These cristae, right? And the gel-like substance filled inside that is called matrix, right? And inside the matrix there are ribosome, function of ribosome, formation of protein. But yes, the size of ribosome is smaller in like bacteria. So they also have 70S type of ribosome. Okay. Inside mitochondria, there is a small circular DNA called mitochondrial DNA, right? There is a mitochondrial DNA and this is circular DNA, right? It resembles with the bacteria. Okay, it resembles with the bacterial layer. Now, here I have tried to enlarge the cristae, right? Look, this is the cristae, right? At the surface of cristae, there are some lollipop kind of structure or tennis racket like structure. They are called oxysome. So you can see, look, again, I have enlarged the oxysome. Oxysome is made up of two parts. The front head, this globular F1 particle and base F0 particle. What happened? ATP, ADP, adenosine diphosphate, ADP and phosphate comes over here and they produces ATP, right? Mean the synthesis of ATP takes place in F1 particle or oxysomes. What are the oxysomes? They are found on the surface of cristae, right? That's how the formation of energy takes place. Formation of ATP is called oxidative phosphorylation. The production of ATP is known as, this process is called oxidative phosphorylation. Tell me, is it clear or not?
Yes, sir.
So the diagram is a bit complex. Do you want to write down right now or after the class? We'll do it. Because it will take time. I know. For you, it may be a bit difficult.
So, sir, we should leave place and then start writing.
Right. That's a smart idea. Leave the space for that. Take the screenshot of this one, right? But draw it today only, right?
Okay,
Don't
sir.
skip it. Okay, Martin? Right. So start the writing down about the mitochondria. Who discovered the mitochondria? Who discovered the mitochondria? This is important question. Basically, mitochondria are first observed by, give me 1 second. Yes. Okay, so start writing now about the mitochondria. So I was telling that the mitochondria was first. Mitochondria are first observed by Kolliker. C. Kolliker. His name was Kolliker, right? So Kolliker. Kolliker in 1880. 1880, right? So Kolliker observed, right? Who given the name mitochondria, right? Mitochondria name was coined by Venda, C. Venda. His name was C. Venda. So C. Venda was the person. Venda, right? So Venda given the name of mitochondria in 1897. Some places I have seen there is a mistake. It is written that C. Venda discovered. No. C. Venda did not discover. C. Venda basically given the name of mitochondria. It was discovered by Kolliker only. That's what you have to remember. Okay. So please start noting now. C. Venda given the name mitochondria. Mitochondria are found in all aerobic eukaryote. Mitochondria are found in all aerobic eukaryote. Full stop. Next point. They are absent in prokaryotic cell. They are absent in prokaryotic cell and anaerobic, mind it, underline, and anaerobic eukaryote. Full stop. They are also absent in RBC. They are also absent in RBC. Next point. The size of mitochondria is 0.2 to 1 micrometer, 0.2 to 1 micrometer in diameter and 1 to 4.1 micrometer in length. Next point. They are double membrane structure. Full stop. The next one. The gel-like substance filled inside mitochondria, the gel-like substance filled inside mitochondria is called matrix. Is called matrix. Full stop. Mitochondria have single circular DNA called mitochondrial DNA. In bracket, please wrote down mtDNA. Called mitochondrial DNA, which resemble with bacterial DNA. Next point. The inner membrane have finger-like structure. The inner membrane have finger-like structure called cristae. In same point, right? Called cristae. Full stop. At the surface of cristae, they have oxysomes. Full stop. Next point. Oxysomes are made up of F1. Oxysomes are made up of F1 and F0 particle, which are site for production of ATP. Full stop. The formation of ATP from ADP. Formation of ATP from ADP is called oxidative phosphorylation. Oxidative phosphorylation. Full stop. Next point. Mitochondria have 70S ribosome. Mitochondria have 70S ribosome like bacteria. Next point. The Krebs cycle, that is burning, the process of burning of glucose called Krebs cycle. We'll discuss that in plant physiology part, right? Krebs cycle goes on in matrix. Krebs cycle goes on in matrix. Is that clear? Everything clear?
Yes, sir.
Okay, great. So one last thing, the formation of new mitochondria takes place. Formation of new mitochondria takes place by division of pre-existing pre-existing mitochondria. Is that clear?
So,
Fine. So please draw the diagram after the class. Leave the space for that. The next one is plastid. So basically we have to study the chloroplast part, right? So first let me tell you what are the plastids, how many type of the plastids are found. You have to note down only that part which I am telling you, not everything. So basically plastids are found in plant. They are again they are called proplastid, right? From proplastid, formation of plastid takes place. Formation of three kind of the plastid takes place. Proplastid convert these three kind of the plastic. First, chromoplast. They provide color to flowers. They provide color to fruits, right? So they have inside them there is they remains filled with some pigment, right? And they provide color to the except green color. They provide color to flower, right, and fruits, right? Like apple is red outside from outside, means the cells have the chromoplast, right? Second one is chloroplast. Chloroplast is green color and it is found in the leaves, green leaves, and its function is photosynthesis. Third one is leucoplast. Leucoplast are found in the those part which store the food, and their function is storage of food and synthesis of some fatty acid. Right now, further, uh, because this leucoplast store the food, means like in the potato, the food is stored in the form of starch, so leucoplast store the starch. Right. So that is the function. They are colorless. They do not have any color. The leucoplast maybe if it store the sugar or starch, this is called amyloplast. If we store the oil or lipid, it's called elaioplast. If we store the protein, this is called proteoplast, right? So they are the type of the leucoplast. Is that clear? Clear? Tell me, guys. Clear or not? Ram and Martin, what happened?
Yes, sir.
So please write down quickly. First, write down plastid and make the arrow and plastid out of three types. First, second, third. Then amylo, elio, and proteoplast in front of leucoplast.
okay, sir.
Done.
Sir.
Okay. Martin, done.
Yes, sir.
Okay, so first point. The term plastid was given by Ernest Haeckel, E. Haeckel. Full stop. Next point. Formation of plastids takes place from pre-plastids or proplastids. Next point. Chromoplast store pigment other than green. Chromoplast store pigment other than green. Full stop. They provide color to flower and leaves. Flower, fruits, and colored leaves. Next point. Chloroplast store green pigment, chlorophyll. Chloroplast store green pigment, chlorophyll. Full stop. They provide green color to plant and perform photosynthesis. Next point. The leucoplast store food and they are colorless. Full stop. They are found in food storing part. Now the main heading is chloroplast, right? Again, you have to do the same thing. You have to leave the space for your diagram, right? So look, this is the chloroplast. You know, they are responsible for these structures are responsible for photosynthesis. They are found in all green part of the plant, irrespective of leaf, stem, whatever the thing is green. Again, like mitochondria, they resemble with the mitochondria. They are also double membrane structure, outer membrane and inner membrane, right? Inside the membrane, there is a gel-like substance. In mitochondria, we call that matrix. Here we call them stroma, right? Stroma. Inside this gel-like substance, stroma, there are some structure. These structures are called granum, right? These structures are called granum, right? every granum is made up of a small saucer-like structure, or we can say biconvex this structure. So these structures, every granum is made up of 40 to 60 these structure, and these structures are called thylakoids, right? What we call them? Thylakoids. Sorry, granum. I'm sorry. The chloroplast may have 40 to 60 grana, right? And every grana is made up of the structure called thylakoid. Every grana is made up of 2 to 100 thylakoids, right? Inside thylakoid, there is a substance, there is a pigment called chlorophyll. Chlorophyll is magnesium compound. That is very important. Chlorophyll is magnesium compound. Like the mitochondria, it also have its own circular DNA, right? This is called chloroplast DNA, right? Please note down these some points, right? Please note down. Like CLPT DNA, chloroplast, please note down this one, right? Note down this structure, this. Just do one thing. Note down a granum and thylakoid. Then after thylakoid, chlorophyll. That thing, please you note down. Rest, you can draw the whole diagram after the class. But this thing you need to note down. And I will dictate it next class because we are running short with the time. Time's not left. So in the meantime, you can note down this thing, this granum, do not a single granum, then this thylakoid, and write out granum maybe 40 to 60, right? And thylakoid maybe 2 to 100. And inside thylakoid, there is a chlorophyll and magnesium. Only 2 or 3 cell organelles are left, like chloroplast will dictate in next class, ribosome and cytoskeleton and nucleus. That's it, right? Only 3 or 4 things. Means in next class, we'll finish the cell biology part, right? I mean, on Thursday.